Hi everyone. Today we're going to dive into the most important balancing acts happening in every single one of our cells, the regulation of glycolysis and gluconeogenesis. Now I know those are big words and sound intimidating, but stick with me. Glycolysis and gluconeogenesis basically control whether your body is spending or saving energy. And right in the middle of this balance is an enzyme called aldolase, kind of the unsung hero that keeps both of the sides talking to each other. We're also going to talk about how the enzyme is regulated by transcription factors like HNF1, HNF3, CEPB, and DBP, all of which act like managers that tell your liver when to ramp things up or slow things down. So think of this as exploring how your body runs on its own biochemical economy, deciding when to earn energy and when to store it. All right, let's start with glycolysis. This is your body's go-to way to make quick energy. It happens in the cytoplasm, right in the cell, and it takes one molecule of glucose and breaks it down into two molecules of pyruvate. Now, the cool thing is that it doesn't require oxygen, so this process can happen even when your muscles are working hard, like during a sprint or a workout. You get two ATP, that's your energy currency, and two NADH molecules, which later help generate even more energy. There are a few key enzymes that act like control switches: hexokinase, phosphofructokinase 1 or PFK1, and pyruvate kinase. These are the spots where the pathway can speed up or slow down, depending on how much energy the cell already has. So when ATP is high, meaning you've got plenty of energy, glycolysis slows down. When ATP is low and AMP goes up, that's the cell's way of saying, hey, we're low on fuel, let's crank it up. It's a really cool and efficient feedback system. Now, flip side, gluconeogenesis. This one's the body's backup plan. It happens mostly in the liver and a bit in the kidneys, and it basically rebuilds glucose when we're running low, like fasting or between meals. Instead of breaking glucose down, gluconeogenesis takes smaller molecules, things like lactate from muscle, glycerol from fat, and amino acids from protein breakdown, and rebuilds glucose from scratch. But here's the thing, it's not just a reverse of glycolysis. Some steps of glycolysis are irreversible. So gluconeogenesis has to take some detours using different enzymes. These are glucose 6-phosphate, fructose 1,6-biphosphate, and PEP carboxykinase. And yes, it costs energy, 6 ATP per glucose molecule, but it's worth it because it keeps your blood sugar stable when you haven't eaten. Think of it as your biochemical emergency generator kicking on when fuel levels drop. Now, this part is fascinating. Your body never runs glycolysis and gluconeogenesis at full speed at the same time. That would be like pressing the gas and the brake on your car at once. To avoid that, the body uses allosteric and hormonal regulation. When ATP and citrate are high, glycolysis slows down because energy is plentiful. When AMP is high, that's the signal to speed things up. There's also a special molecule called fructose 2,6-biphosphate. This one is the real decision maker. It tells the cell, hey, let's focus on glycolysis right now, or switch gears, we need to make glucose again. And hormones control this on a big picture level. So insulin, which comes out after a meal, tells the liver to store and use energy, boosting glycolysis. Glucagon, released during fasting, tells the liver to make glucose instead. So you've got both local enzyme regulation and full body hormonal signals, keeping everything balanced. Okay, aldolase. This enzyme is the bridge between glycolysis and gluconeogenesis. It's like that one employee who somehow works in both departments. In glycolysis, aldolase takes a 6-carbon molecule, fructose 1,6-biphosphate, and splits it into two 3-carbon molecules. So glyceraldehyde, 3-phosphate, or G3P, and dihydroxyacetone. Dihydroxyacetone. Phosphate. There we go. DHAP. But what's amazing is that it can also run the reaction backwards. Hence this double arrow that we see over there. During gluconeogenesis, it links these two trioses back together to rebuild fructose 1,6-biphosphate. So depending on whether your body is burning or making energy, the same enzyme just changes direction. Now, aldolase actually comes in 3 different versions, or isoforms, each designed for a specific job. Aldolase A is found mostly in the muscle and red blood cells, tissues that burn through energy quickly and rely on glycolysis almost constantly. Aldolase B is found in the liver, kidneys, and small intestine. This one's special because it can handle both glycolysis and gluconeogenesis, and it also breaks down fructose. Aldolase C is found in the brain, where neurons need a steady flow of energy. All 3 isoforms look similar structurally, but they're tuned differently. It's like having 3 versions of the same tool, one for speed, one for precision, and one for endurance. So even though all aldolases share a similar overall shape and use what's called a skiff-based mechanism, their small differences make them uniquely suited for different jobs. For example, aldolase B can process fructose 1-phosphate, something that others can't do efficiently. That's really key for the liver since it's the main site of fructose metabolism. Muscle aldolase A, on the other hand, works fast. It needs to because your muscles demand energy instantly. Aldolase B works slower but is versatile. It can go in either direction depending on your body's needs. So that fructose connection that I referred to earlier. So when you eat fruit or anything sweetened with fructose, your liver takes over processing it. Fructokinase turns fructose into fructose 1-phosphate, and then aldolase B splits it into DHAP and glyceraldehyde. These pieces can jump into glycolysis or gluconeogenesis depending on whether you just ate or you're fasting. But if someone has a mutation in the ALDOB or ALDO gene, the enzyme doesn't work right. That leads to hereditary fructose intolerance, where fructose 1-phosphate builds up, sucking up the phosphate and stopping ATP production. Clinically, that's pretty serious. It can cause hypoglycemia, vomiting, and even liver failure if fructose keeps being consumed. This is a classic example of how a small enzyme defect can have a system-wide effect. Okay, so how does the body decide when to make more aldolase B? That happens through gene regulation in the liver. The ALDOB gene has promoter regions that respond to transcription factors. These are proteins that turn genes on and off. Specifically, HNF1 here and HNF3, which here's a beta, are like the main managers ensuring that aldolase B is only made in liver cells. And then we have CEBP, which is here. Adjust the amount based on what's going on metabolically, like fasting or feeding. And finally, we have DBP, which is here. Oh, and there's another CEBP. DBP acts like the timekeeper, syncing the enzyme's production with your circadian rhythm, meaning you actually make more autolyse B at night when gluconeogenesis tends to increase. It's a real neat example of how metabolism, hormones, and even your internal clock all talk to one another. HNF1 and HNF3, or hepatocyte nuclear factors, are the backbone of liver-specific gene expression. They control a lot of key metabolic genes, including autolyse B. They bind to specific DNA sequences in genes promoter, and recruit other helper proteins to get that transcription started. When these factors are mutated, they can cause disorders like MODY, or maturity onset diabetes of the young, because glucose regulation goes haywire. In the context of autolyse B, these transcription factors make sure that the enzyme is made when the liver needs it, for example, after a meal or during periods of active metabolism. Now, CEBP and DBP are more fine-tuning. So CEBP reacts to hormones when glucogen is high during fasting and increase autolyse B production to support gluconeogenesis. DBP as a circadian layer, meaning autolyse B levels fluctuate throughout the day. So you actually get higher levels overnight, which matches the liver's increased glucose production while you're asleep. So when we step back and look at this together, the hormones, the transcription factors, and the circadian rhythm, it's clear how tightly controlled the system is. Insulin and glucagon provide the big picture hormonal signals. HNF, CEBP, and DBP translate those signals into gene level changes, and everything runs on a daily schedule. This coordinated system prevents chaos, it keeps blood sugar stable, balances energy, and makes sure the enzymes like autolyse B are always available at just the right levels. So why does this matter? Because when autolyse B regulation goes wrong, we see it in disease. In the hereditary fructose intolerance that I mentioned earlier, the enzyme doesn't work. So fructose can't be processed properly, leading to phosphate depletion, ATP loss, and hypoglycemia. On the other side, too much autolyse B activity or misregulation has been linked to insulin resistance and metabolic syndrome. It's a reminder that our biochemistry is all about balance. Too much or too little of even one enzyme can disrupt the whole system. All right, to wrap everything up, glycolysis and gluconeogenesis are two key pathways that keep our energy system in balance. Autolyse acts as the bridge connecting them, with autolyse B, in particular, playing a unique role in both energy and fructose metabolism. Its expression is fine-tuned through transcriptional control with HNF, CEBP, and DBP, all working together to match enzyme levels with our body's needs. When these systems are working properly, everything runs smoothly. But if the regulation fails, we see disorders like hypoglycemia and metabolic inflexibility. So this one enzyme really shows how interconnected metabolism truly is. Thank you for listening and following along today. This is really one of the most fascinating examples of how organized our metabolism is. So before I close, take a second to think about how your own body balances between energy use and storage. It's literally happening every moment. Any questions or thoughts about what stood out to you the most? Maybe which regulatory mechanism makes the most sense, hormonal, enzymatic, or genetic? I'd love to hear about any questions or connections that you're making. Thank you.